A controversial theory suggests that life began when RNA spontaneously started self-copying, and now researchers claim to have partially recreated that process in a laboratory. Could this be artificial life? In interviews with the Washington Post, WIPO, scientists say they have created an RNA molecule that made copies of other types of RNA, bringing experts closer to creating conditions for early life on Earth in a lab. Scientists at the Salk Institute for Biological Studies, USA, worked on the basis of the theory that before DNA or proteins existed, RNA existed as the initial ingredient in the so-called primordial soup. Have researchers succeeded in creating artificial life? In their research, according to WIPO, they created a laboratory-made RNA molecule that accurately copied other molecules and resulted in a functional enzyme. Now that the Institute has taken this step, it is ready to study the early evolutionary stages of life in unprecedented ways. Gerald Joyce, the president of Salk, who is a co-author of a new study published in the Proceedings of the National Academy of Sciences, told WIPO that although the molecule made in the lab by researchers still does not self-copy, the one they created represents a huge step towards creating artificial life in the laboratory. If the created RNA is capable of self-copying, Salk's president said, then it would be alive. How does the process work? As WIPO explains, RNA must make copies extremely similar to the original for Darwinian evolution to occur. If something goes wrong, things deteriorate rapidly. And just like with an old and faulty photocopier, each subsequent copy becomes blurrier until it is no longer clear what the original source material was. If the error rate is too high, you can't maintain genetic information. It just blows up, explained the Salk president. However, exact copying of RNA doesn't work either because it doesn't provide the types of mutations that promote growth. To achieve just the right amount of deviation, Joyce and his team created an RNA that makes copies of what is called hammerhead RNA, which cuts molecules. When the copying molecule does what it needs to on the hammerhead RNA, each new generation, according to WIPO, could also cut, and each subsequent generation becomes even better at copying. This new threshold is described by John Chaput, a professor of pharmaceutical sciences at the University of California at Irvine, USA, is monumental. At first, we looked at everything as something amazing. It's very interesting, said Chaput, who was not involved in the research. This is fascinating research, although if Salk or collaborators succeed in creating artificial life in the laboratory, it will certainly raise new and urgent ethical questions about synthetic life forms.